Welcome all. Today we are discussing on the topic fluorescence resonance energy transfer. FRET is a physical phenomenon whereby energy created by fluorescence excitation of one molecule is transferred to an adjacent molecule. It is a non-radioactive process whereby energy from an excited molecule, that is the donor, is transferred to a second non-excited fluorophore, that is an acceptor. FRET is used as a molecular ruler and the donor is usually directly excited, whereas direct excitation of the acceptor should be avoided. The phenomenon FRET must be satisfy five criteria. First one, the emission spectra of the donor must significantly overlap the excitation spectra of the acceptor. Second one, donor and acceptor must be aligned. Third, the donor or acceptor separation can be not greater than 10 nanometer. Fourth one, the donor has a high quantum yield. And the fifth one is donor is saturated by the acceptor. Assuming that these criteria have all been met, then energy transfer will occur and is described by the Forster equation. And there are generally three commonly used approaches for measuring FRET: decreased donor lifetime, decrease in donor emission intensity, and increase in acceptor emission intensity. Next, we are moving on to some factors that can affect FRET. First one, brightness of fluorophore. It is best to use a donor and acceptor that are of comparable brightness. Second one, fluorophore alignment. It is particularly important to ensure that the fluorophore tagged antibodies do not bind epitopes that are at the far end of each protein. Third one, spectral crosstalk. As some degree of spectral overlap between the donor and acceptor is necessary for FRET, spectral crosstalk will inevitably occur for any FRET pair and must be addressed when FRET is measured. And the last one is donor or acceptor stoichiometry. If the donor to acceptor stoichiometry is outside the range of 1:10, the FRET signal may be marked by the background of fluorescent labels that are not undergoing FRET. Next is applications of FRET. FRET is used in protein studies. Protein partners can be co-localized by FRET. In biological membrane mapping, in cell biology, FRET is used to study metabolic and signaling pathways and is used to study DNA mutations, etc. Advantages of FRET. It is relatively cheap to implement. Measurements are obtained very rapidly, very good at measuring changes in distance. And the disadvantages of FRET include: it only works if fluorophores are in correct orientation. CISO probes can introduce problems, gives no information about which probe moves, can be pH sensitive. Thank you.